So our topic today is the origins of disease. Next week I'll be talking more about the immune system in particular. Today's talk will be an evolutionary perspective on disease to a large extent. This is a topic which I think I'm fortunate to be able to present in that it's much more accessible than topics that deal with objects that are either tens of billions of meters in size or 10 billionths of meters in size. So we're going to be talking about issues that confront all of us sooner or later. I think that's fair to say. And so first I'm going to introduce some basic concepts necessary for understanding any biomedical subject. We're going to talk about the fundamental biological principles that apply to organisms of all sorts, a little bit about cells in particular, which are critical for understanding anything that happens in the body and in the context of understanding diseases. Then we're going to talk a little bit about genes, genotypes, and phenotypes, which are terms I'll be defining for you, although I'm sure many of you have heard these terms and have some idea what they mean. Then we're going to talk about diseases and how they come about, the origins of diseases from an evolutionary perspective. We're going to define diseases, address some of the evolutionary factors that influence disease susceptibility, and then talk about the fact that disease and how to interpret the importance of a disease sometimes depends on the context. And then the last section is going to focus on a concept which I think is really quite significant, especially in the era of what they now call genomic medicine or personalized medicine. And that's the concept of heritability. I'm going to define the term for you, describe why the term is relevant to evolution, and then discuss why it's relevant to talking about diseases and disease susceptibility. And then finally, I'm going to describe some of the myths and misperceptions regarding this concept of heritability. So let's get to the basic principles of life sciences. All organisms are composed of cells. So that is the fundamental biological subunit, you might say, of which all organisms are composed. Now, cells are the smallest units that are unambiguously alive. So one could argue about whether viruses are alive or not, and equally eminent biologists differ on that question, and I think there are some interesting ways to look at it, but we're not going to focus on that today, unless someone's interested later. But cells are clearly alive if you put them into little plastic containers in what we call tissue culture, in other words, outside the body, and put them in an incubator with the right temperature and the right gas content, they'll grow and they'll reproduce, and you'll end up with more and more of them as long as you keep feeding them and taking away the waste products. So they're clearly alive, whereas molecules are not clearly alive. Cells, however, are composed of molecules, and molecules are simply assemblages of atoms. You've already learned from the talk last week and earlier talks by Glenn Starkman and Cyrus that each of the particles, when you smash it with some other particles, tends to have smaller components. And this is true in biology. There's a hierarchy of organization and complexity, you might say. There are small molecules, so-called, like adenine triphosphate, which is involved in energy exchange in the cell. There are what are called macromolecules. These are molecules composed of many, many subunits, such as amino acids or nucleotides, like DNA and RNA, which are composed of nucleotides, proteins composed of amino acids, polysaccharides, which are composed of monosaccharides, et cetera, and lipids. These are larger molecules. And they go by the name macromolecules in the context of biology. And then there are complexes of molecules, entities like ribosomes, which are assemblages of protein and RNA in the cell on which new proteins are made. But they're much more complex than any one protein or any one RNA molecule. And then the cells contain structures of intermediate sizes and degrees of complexity, so-called organelles. And I'll just briefly mention a few of those, like mitochondria, for instance, or lysosomes. And then just to give you a little bit of context overall, there are three domains of organisms, at least many people believe that's a reasonable description of what is in fact a very complex reality. Bacteria, archaea, which are sort of like bacteria, but they're also sort of like our kinds of cells. They're often found in very inhospitable environments like thermal vents in the bottom of the ocean and so forth. And they are often adapted to conditions which are really quite remarkable. And then eukaryotes is a broad term which covers all plants and all animals and fungi. And so we are actually considered part of the group of organisms which includes plants and fungi as being more closely related to one another than we are to bacteria, for instance. Now let's talk about human cells. Now this is just a diagrammatic, schematic illustration of a cell. It's a sort of a generalized cell. It's not any particular cell. And you can see that there's quite a bit of structure there. This purplish area in the middle here is the nucleus. I guess it's actually more like magenta. And then the darker region in the middle of the nucleus is called the nucleolus. And we're not going to get into the specific functions of the nucleolus today, but that's where the chromosomes and the genes reside. And we're certainly going to be talking about genes. And then you can see there are a lot of other structures. This is what's called the endoplasmic reticulum. You don't have to worry about remembering these names, but that's a structure which is bound by membranes, which has to do with the synthesis and distribution of proteins, which are crucial for all the functions of a cell. This reddish blob over here is called the Golgi apparatus. It's also involved in that process of trafficking proteins, distributing proteins to where they need to go. You see a mitochondrion. That is the energy powerhouse of the cell. It's sort of like a generator or a power plant, you might say, for the cell. The point is that there are a lot of different structures, and different cell types have different arrays of these structures, although almost every cell has mitochondria and so forth. So let's talk about some of the key characteristics. All cells have a lipid membrane. So lipid is the same as fat. And it's also often called the plasma membrane. These are highly structured entities, that is cells. It's actually more like a gel inside the cell than an aqueous solution, a water solution. So it used to be thought many, many years ago that inside the cell was like a bag of enzymes. Enzymes are proteins that have a particular kind of biochemical activity, catalytic activity. They make reactions happen more quickly. And it turned out that when you look at cells, they're actually highly structured. And it's more like Jell-O than like a glass of water. And there are many compartments in the cell. I've already mentioned the nucleus, and outside the nucleus is the cytoplasm. And then within the cytoplasm, you have all these other structures: the endoplasmic reticulum, the Golgi apparatus, the mitochondria, the lysosomes, et cetera. And every cell, with the exception of red blood cells, also known as erythrocytes, or platelets, which are involved in blood clotting, every cell in your body has a nucleus. And in some cases, there's more than one. And that's where, again, where the genes are. And again, there are all these other internal structures which I've already alluded to in the schematic diagram. Now here's some examples of human cells. I've chosen particularly cells from the immune system because we will be talking about immune system function next week. And you can see that they're quite different looking when they're stained by various dyes, and that's what we use as a tool all the time in the pathology department. So this is a small lymphocyte. That's one of the basic white blood cells that's involved in mediating immunity. This is a plasma cell. This is a type of lymphoid cell that makes antibodies, which are, again, involved in immune function. Dendritic cells, which were actually the discovery of these cells was actually the basis of the Nobel Prize this past year. Ralph Steinman was the recipient of that prize. You may know he was the Nobel laureate who died 3 days before the prize was awarded. And then that's a mast cell, which some of you who have allergies may have heard of because it's involved in the causation of your symptoms. Now, we have to make a couple definitions right up front, which are very crucial to much of the rest of what I'll be discussing. The genotype is the genetic endowment of an individual. So that would be all the different genes in your genome if you're talking broadly. In some cases, when you talk about genotype, you may be focusing on just a particular few genes or a single part of the genome. In contrast to genotype, which is about what genes you've inherited, the phenotype is the constellation of observable traits. And traits can be defined at all different levels, from behaviors to morphological features, and other structural features, to molecular features. So these are two fundamental concepts: your genotype, which is based on exactly what genes you have inherited, and your phenotype, which is how you act in the world. What are your features? What are your attributes? So let's talk a little bit about genes, genotypes, and phenotypes. Human genes, like all genes, are sequences of deoxyribonucleotides. Now, there are viral genes which are ribonucleotides. They're made of RNA instead of DNA. But basically, genes are DNA. There are multiple definitions of genes. So you will hear people talk about genes, and sometimes they might, for instance, mention that when the Genome Project was being finished up, the Human Genome Project, different groups, there was a federally funded international group that was funded by governments, and there was a private group. The government-sponsored group was led in the United States by Francis Collins, and some of you may know that the privately financed group was run by Craig Venter. And they both came up with somewhere in the vicinity of 20 to 30,000 genes at the end, but they didn't agree on what those genes were. Largely their lists overlap, but not completely. And that brought up the issue that they weren't defining gene exactly the same. And it turns out there's different aspects of genes that are emphasized in different contexts, like mutation, susceptibility to mutation, susceptibility to recombination, which is when genes exchange locations on chromosomes, and actually determining a trait. So I just want you to be aware of that, because that can confuse people at times. Genes reside on chromosomes in the cell nucleus, and it turns out the mitochondria also have their own genes. So all of our genes aren't in the nucleus. Some of them are in our mitochondria, which interestingly you inherit only from your mother. And human cells contain 46 chromosomes. Now it's interesting, it actually for many decades the official number was 48, even though there are only 46. And so people would count the chromosomes when they did an analysis of a particular set of cells, and because they were expecting to find 48, that's what they found. But in actual fact, there's 46. Now here's two other key terms. Positions on chromosomes are known as loci. The singular is locus. So a locus is a specific chromosomal location where a gene resides. It's like an address. So we could talk about your location on a street, and that would be a locus. Alleles are alternative nucleotide sequences residing at the same locus. Now, humans are diploid. You have two chromosomes of each type. Like chromosome 1, you have one from your mother, one from your father. The identities of two alleles at a locus determine the genotype, and a phenotype is a cellular or organismal trait, as mentioned. Phenotypes result from the interactions between genes and environmental factors. So here's an illustration of a maternal and a paternal chromosome 6. You have 3 loci I've indicated. Those are just the names of loci which happen to be relevant in immunology. And the group of these 3 alleles together at their respective loci. So there are different alleles at A from the mother and A from the father. This could be A0201. That's just the terminology we use. And this could be A2301. This is one haplotype, and that's another haplotype, just so you are familiar with that term. And this is the way the transmission works with a simple Mendelian genetic situation. So the female, the mother, has 2 haplotypes. The father has 2 haplotypes. And all 4 possible combinations can occur. So the chances of any 2 siblings being identical for these particular haplotypes is 1 in 4. Now here's what's called the central dogma of biology. The DNA, information in DNA in the form of nucleotide sequences, is transcribed into RNA, which are ribonucleotide instead of deoxyribonucleotide sequences. And the RNA then codes for proteins, which are made up of amino acids. There are only 4 nucleotides in DNA and RNA, and there are about 20 naturally occurring amino acids in proteins. And so in understanding genotype and phenotype, a triplet of nucleotides gives rise to a single amino acid. That's called a codon. And here we have a specific example. AAA in the DNA gives rise to UUU in the RNA. These are called complementary bases. And this process is called transcription. And this process from RNA to protein is called translation. And that's an amino acid. That's one naturally occurring amino acid, phenylalanine. So transcription schematized, you have two strands of DNA. One is the strand upon which the RNA is made in the transcription process. The other strand is complementary. So the strand up here, where each of these little ladder rungs corresponds to a nucleotide, is identical in sequence to the RNA. We're going to have to stop and take some questions in a second. I just want to make a couple last points. So genes lead to polypeptides. And this is the translation process schematized, where another kind of RNA brings in amino acids to create a growing polypeptide chain. And the bottom line is that genes give rise to traits, but they're not the only factors which lead to traits.
Following the migration out of Africa, and they're doing all that using scrapings from your mouth or whatever, and they're using the mitochondria for that. Is it really that reliable to be able to tell the migration by the mitochondria from the mother?
The mitochondrial genes can give you information on the maternal line for that individual. And it's one method which has been very useful in mapping out migrations. And it's not an area where I would say I have expertise, but I think that analysis of mitochondrial genes, as well as genes from the nucleus, the autosomal genes, has been extremely valuable in giving anthropologists a sense of where populations have migrated. So yeah, I think it is a very effective means.
There was a discussion on NPR yesterday, I believe, about bacteria comprising most of the dominant part of the organisms in the body.
So yes, it's true that there are probably 10 times more bacterial cells in a human body, counting the bacteria in your gut, the bacteria on your skin, and selected other regions. They're not everywhere, although some people say things like that. But there are a vast number of bacterial cells in your body, and there are many, many bacterial species, probably on the order of 1,000. And in addition to having 10 times more bacterial cells than human cells, so-called, in a human being, there are also many more bacterial genes than human genes in terms of different nucleotide sequences. So we are an ecosystem, every one of us.
Why is there confusion or disagreement about the smallest living unit?
Because you end up having to decide where you draw the line between living and non-living. So a virus is not a cell. It doesn't have a metabolism, per se, but it replicates in a manner dependent on cells that it infects. It evolves, and it has genes which in some cases appear to be derived from eukaryotic cells. So while viruses are more like, yesterday on NPR there was an interview with a science writer who said, in response to a question from his interviewer about bacteria and viruses and let's say eukaryotic organisms or cells, what are the similarity relationships there? And the science writer said that viruses that human cells are more like bacteria than they're like viruses. Well, in some ways, yes, but in other ways, no. And different specialists focus on one aspect or the other. So some people say viruses are alive, and some people say they're not because they don't have a metabolism. So just to quickly finish up about genotypes and phenotypes, I'll put these up all at once. It's a complex relationship between the genotype and the phenotype. And we don't have the time this evening in a single hour to explain all of these interactions and processes. But some of these will become apparent as we go through the rest of the talk. Because we're diploid, we have two copies of every locus, and that means we can have an allele from the mother and an allele from the father, and they can sometimes be the same, and that's called homozygous, but sometimes they're different, and that's called heterozygous. And sometimes the impact of a particular allele depends on what other allele it is paired with at a particular locus. In addition, Sometimes the influence of a particular allele at locus 1 depends on alleles at locus 2 or other loci scattered throughout the genome. So there are gene-gene interactions, and that goes by the name epistasis. There are, of course, important gene-environment interactions, and I'm going to give you a rather striking example of that a little bit later. There are random effects on gene expression, which I will show you an example of momentarily. Each gene, in many cases, has more than one impact on the body, and that's called pleiotropy. So changing a gene can have effects in many different respects. There's a mutation. And multiple genes can contribute to individual traits. So there isn't generally a single gene, single trait, simple relationship. That would be easy and nice, but it's not, in fact, what usually obtains. So here's a couple of examples. This is a picture of the first cloned cat and its parent. This pointer is not working too well. So there's the offspring, named CC for carbon copy. I thought this was a missed opportunity. I would have called it copycat, but and the mother or parent or whatever you want to call it, the cat from which CC was derived is called Rainbow. And you'll notice, although they look pretty similar, the coat pattern, the coat color pattern, is not identical. And this is just to make the point, and we could find other examples, that even though they are putatively genetically identical, they are not phenotypically identical. So genotypic identity does not guarantee phenotypic identity. Now here's an example of clones, genetically identical bacterial cells, E. coli, just like the bacteria in your gut, one of the main species in your gut, or my gut, nothing personal there. Each of these bacteria is engineered to express two different proteins which express different colors fluorescently. And if they're not looking the same color, that means they have different gene expression magnitudes of these two different genes than their neighbor. And so I won't go into all the complexities of the experimental design, but in this panel, the upper left panel, A and D and C and F, you see varying shades of green and orange. And that tells you that these genetically identical cells are differentially expressing the same genes. And so the phenotypes are not identical, even though the genotypes are identical. So we already did that. So now let's go on to disease and what disease is. So there are a couple definitions I just pulled from my original medical school medical dictionary. You can tell because it's from 1972, which dates me. So this is not a concept you're unfamiliar with, but I just thought I'd give you a formal definition. So according to the medical dictionary, a disease is an illness, sickness, an interruption, cessation, or disorder of body functions, systems, or organs. We've all, I hope you're feeling okay. We've all experienced disruptions of bodily function at one time or another. And if you haven't, please raise your hand. We should probably sequence your genome. Or it can refer to a specific disease entity, meaning an entity that meets the following criteria, or at least two of these criteria. A recognized etiologic agent. Etiologic agent means the source, the cause of the disease. An identifiable group of signs and symptoms, or a consistent anatomical alteration. Now, there are many aspects of disease that are of interest to people who focus on disease, pathologists, and I'm a clinical pathologist, which means I run a laboratory, as opposed to an anatomic pathologist who does biopsies and autopsies and that sort of activity. Etiology is about the cause of a disease, as opposed to etymology, which is about the origin of a word. Pathogenesis is focused on the mechanism by which the disease develops. So for instance, if I tell you that Mycobacterium tuberculosis is the etiologic agent of tuberculosis, that tells you a key cause, although it's not really the only cause. But it doesn't tell you how the disease, the manifestations of that infection come about in an individual. That would be the pathogenesis. Then there are the molecular and morphological changes, and that's really what pathologists focus on. I actually have a typo there. The morphologic changes are what anatomic pathologists focus on. When you do an autopsy, you're looking at gross morphological changes in organs and tissues. And when you do microscopic analysis, you're looking at histopathologic changes in morphology. Clinical pathology is more focused on molecular changes, although now anatomic pathology also can assess molecular changes to some extent. And then there are the clinical manifestations, which is what all of us know about from direct experience and what generally takes us to see a physician. Now, the historical origin of the study of disease, although it's always dangerous to attribute any great advance to a single individual, if you had to pick one individual, this individual, Giovanni Battista Morgagni, would certainly merit a great deal of attention. He was born in 1682 in Italy, and he studied medicine in Bologna, which was one of the original great medical centers, medical schools, historically, with a guy named Antonio Maria Valsalva, who's also a famous person in terms of his contributions to medicine. And he developed what we now call the anatomic concept of disease. Now, it may stun you to learn that before this point, in the 1600s, the 17th century, most concepts of disease causation dealt with very diffuse concepts like miasmas and ill humors. There was no concept that you should correlate a disease with a particular anatomical location in the body. And it was Morgagni's genius to realize that if he followed patients and then studied their bodies afterwards, he could actually correlate changes in their morphology, changes in their tissues and their organs, with the symptoms and signs they displayed when alive. And he wrote this book in 1761 when he published it in 1761 when he was 79 years old, which is pretty amazing to me, called The Seats, at least in English, called The Seats and Causes of Disease as Demonstrated by Anatomy, and the Italian or Latin goes by De Sedibus. And here's a picture of the copy, the front of the book, and this is a likeness, presumably, of Morgagni. Obviously, this is pre-photography. Now, I show you this schematic just to illustrate that ultimately all disease causation, or almost all, involves cells in some way or other. And we're not going to go through this, but the point is, if you have a normal cell and it's exposed to some sort of input, some stimulus, some stress, in many cases the cell can adapt, but if it's pushed too far, then you will have cell injury. In some cases it can be repaired. In other cases it goes on to irreversible injury and cell death. And all disease ultimately results from the dysfunction or the death of cells. Now, there's many ways to classify disease. I don't want to get hung up on this point. There are many ways to classify almost everything in biology. But there are a number of different ways of doing it, and some of these distinctions are worth keeping in mind. One can distinguish between genetic disease and acquired disease. So, for instance, although this is not an absolute distinction, if you inherit a gene for phenylketonuria, you will inevitably have severe symptoms unless your diet is changed to a very unusual diet, which eliminates phenylalanine from the diet. So on the other hand, if you get an infection, that would be an acquired disease. However, I hasten to point out that your susceptibility to infection may be influenced by genes. So again, there's this constant interplay between genes and environmental factors. There are systemic diseases that affect the entire body, like systemic lupus erythematosus, or SLE. Some of you may know people with a disease like that, or a number of other rheumatic diseases that affect the whole body. But then there are organ-specific diseases, like thyroiditis, or pancreatitis. There are infectious and non-infectious diseases, as already alluded to. And there are lots of other categories. Some people talk about inflammatory diseases, metabolic diseases, vascular diseases. But a lot of these categories are overlapping. They don't carve up the universe of diseases into nice, neat little bundles. Now, let's talk a little bit about a Darwinian or evolutionary perspective on disease. The logic of evolution by natural selection is pretty succinctly embodied by this series of statements from Charles Darwin's famous book on the origin of species. As many more individuals of each species are born that can possibly survive, and as consequently there is a frequently recurring struggle for existence, it follows that any being, if it vary however slightly, in any manner profitable to itself, will have a better chance of surviving and thus be naturally selected. And then he goes on to say, although he didn't understand inheritance, from the strong principle of inheritance, any selected variety will tend to propagate its new and modified form. And that's how you get change in a population over time. And Darwinian evolution is about changes in populations over time, not changes in individuals over time. Now, as human beings, we are subject to the same evolutionary factors and processes that every other organism is subject to. We're subject to selection that maximizes fitness, which is defined in terms of your overall genetic success, getting your genes into the next generation and beyond. Greater fitness, where fitness is about getting your genes into the next generation, is not necessarily the same as better health, which is a sobering realization. Forms of inheritance other than genetic may influence human fitness. So what you pass on to your children through cultural means can be just as important in making them fit in an evolutionary sense. So that's another interesting wrinkle or complication. Genetic variants that can promote, let me say that again. Genetic variants that can promote greater fitness at one stage of life may nevertheless lead to lesser fitness at another stage of life. And this goes by the interesting name antagonistic pleiotropy. So this is an example of pleiotropy, where a gene has more than one effect. But in this case, the two effects seem to be at odds, in conflict. So there's a number of studies in humans and other species that tend to find, although it's not uniformly the case, but tend to find that genotypes which favor longevity disfavor fecundity, and vice versa. Now, Randy Nesse, who's a psychiatrist at the University of Michigan, who's very well known in evolutionary medicine circles, has given the following reasons for why we get disease. Why are we susceptible to disease if we're being selected to be fitter? And again, fit in this context has a very technical definition in an evolutionary sense. Pathogens evolve more quickly than humans do. So even though we evolve, we don't evolve as quickly as HIV or influenza virus or pneumococcus. And so sometimes, even though we're evolving and we have some very impressive immune system mechanisms to combat infections, they can get one step ahead of us, or in some cases more than one step ahead of us. Environments change more rapidly than humans do. Genotypes that now are ideal for living in an environment where you're short of food periodically are not necessarily ideal for living in an environment where you can walk into a supermarket and buy, 1 million calories in a few minutes. Evolution is constrained by physical laws and inherent limitations of developmental pathways. Almost uniformly, there are trade-offs in phenotypes. Perfection is generally not a possibility in biology. And reproductive success can be facilitated by traits that undermine long-term health. And useful responses like fever can be taken to excess and then cause medical problems. So there's this huge difference between health, which I define very formally there, and fitness, which again is just the ability to get your genes into the next generation. So if we look at an example of a disease called sickle cell disease, this is a disease you've probably heard of that involves mostly Africans, although not uniformly. And there are two alleles at the key locus, the beta globin locus of hemoglobin. And if you have two parents who are carriers, they have an A allele and an S allele at this locus. They can have 4 kinds of offspring, just like I showed you with the HLA genes. You can have homozygous A, which is normal hemoglobin. You can have two carriers, AS and SA. And you can have someone who's homozygous SS. And it's only these people who have that genotype, SS, who would get the disease. Now, what's really interesting is if you get SS, you get sickle cell disease, which in the absence of medical care, in most cases, although it turns out not in all, is pretty much guaranteed to be fatal at an early age. But if you're a carrier, you're actually more likely to be resistant to falciparum malaria, which is the most lethal form of malaria. There are 4 kinds of human malaria. And so it turns out that the sickle allele is prevalent only in those areas where malaria, falciparum malaria, has been endemic. And this shows you the power of evolution to shape our genetic heritage. Now that's what a sickled cell looks like, and it arises because the hemoglobin, if it's sickle hemoglobin that has the SS genotype, it tends to form these long polymers which distort the cell shape, and that leads to what's called sickling, and that causes all the symptoms, all the manifestations of the disease. I just want to bring to your attention that even in this classical case of a single gene defect, so-called, it turns out that, as this quote says, to some extent the category of complex traits is all-inclusive. Even the simplest genetic disease is complex when looked at closely. Sickle cell anemia is a classic example of a simple Mendelian recessive trait where you need 2 copies of the gene to get the disease. It turns out that individuals carrying identical alleles at the beta globin locus can show markedly different clinical courses. And as it says in the next slide, genetic differences at other loci, one on the X chromosome and one on the autosomes at least, can increase fetal hemoglobin amounts, and that ameliorates the disease.
In what way longevity causes a lesser amount of fecundity?
Okay, so the question is, can I explain more about the trade-off between fecundity and longevity? Most of the studies at this point are really about the correlation, and they haven't completely explored the mechanism. So we can discuss that afterwards. I don't know that there's any single mechanism, especially in different species of organisms, that accounts for this trade-off. But there certainly is some information on that. I don't think we have time to get into it in detail right now. But it's definitely a very interesting area of active research. We'll finish up a little bit about diseases. I just wanted to give you, since we're going to talk about immunology and the immune system next week, I wanted to give you some examples of immune system diseases. So we have allergic diseases, and this first picture on top here is what happens when you have an anaphylactic attack. And this is someone's larynx, not someone who did well, obviously. And you can see that there's almost no passageway there. So this person is asphyxiated. And that could be due to being stung by a bee if you have allergies to bee stings and so forth. Autoimmune diseases. So here we have an example of rheumatoid arthritis, abbreviated RA. And this is someone's hand suffering from rheumatoid arthritis, which has been terribly distorted. And the morphology has been changed. The function has been decreased. And this is an example of how powerful an autoimmune pathology can be. The immune system has the potential to destroy your own tissues if it's not properly regulated. There are cancers of the immune system. You've all heard of leukemias and lymphomas. And there are diseases that result in organ failure necessitating transplantation. And so immunologists have had many major contributions to the studying transplantation biology and the immunology of transplantation. Then we have immunodeficiency diseases. And I don't know how many of you remember. I think everyone in this audience is likely to have heard of the bubble boy back many years ago. This individual named David Vetter was born without an immune system effectively. I mean, it's a little bit of an overstatement, but he had what's called severe combined immunodeficiency, or SCID. He had neither B or T lymphocytes, which are the main 2 types of lymphocytes, although there are many other types. And he eventually got tired of living in a plastic bubble, which kept all those microorganisms away from him that the rest of us have to deal with and manage to handle relatively routinely. And when he came out of the bubble when he was about 12 years old, he did succumb. But he didn't want to live like that anymore. And then we have infectious diseases where the immune system is almost always playing an important role. I just want to close my little section on diseases by talking about this little historical vignette about a very interesting character from Iceland. A guy named Egil, who was the son of someone named Skalla Grim. And he was basically a Viking. And he was famous for Viking raids. And he did what you would expect on Viking raids, pillaging and plundering and some of the other things which are not as wholesome to describe. But it turned out he lived till he was 80. Which is really quite amazing because he had what's called Paget's disease. And Paget's disease is a condition which tends to weaken the bones in most of the body, but it also causes enormous amounts of bone to deposit in the skull. And why that is, I don't fully understand. But this is a skull of someone who died from Paget's disease many, many years ago, not Egil. I don't think we have his skeleton. But it gives you a sense of how thick this skull must have been. And apparently Egil's skull was so thick that he could withstand an axe blow. So if you think about the fact that he had a disease, but that particular disease in that particular context of having routine battles with people wielding axes and swords trying to kill you, in that context, having this particular disease was probably an advantage. not a disadvantage. So the assessment of fitness is not straightforward. And so in this context, Paget's disease was probably a benefit for this, and he lived to 80 if you needed any further proof, if the history is to be believed. So let's go on to defining this concept of heritability. Now this is going to be the most challenging section of this lecture. It's not quite quantum physics, but there is a certain subtlety to it. So heritability, the concept of heritability was introduced to quantify the level of predictability of passage of a biologically interesting phenotype from parent to offspring. So it's really about the correlation between the parental phenotype and the offspring phenotype. Another way to look at it is it's about the extent to which phenotypic variation in a population is due to genotypic variation. Now, I know people don't like to deal with formulas in mathematics, but these are fairly straightforward, and I will explain them to the best of my ability. There's two types of heritability, so-called broad and so-called narrow heritability. Broad sense heritability is the ratio of phenotypic variance due to genetic variation over the total of phenotypic variation. Actually, variance is a statistically, technically defined term. But you can think of it as just variation for our purposes. And so however much variation there is, let's say in height, we can ask how much of that variation is due to genes in some sense, and how much is due to something else, like environment. That's broad sense heritability. Narrow sense heritability, as the name would imply, has a narrower focus. It is only interested in the ratio of phenotypic variance or variation due to what's called additive genetic variation out of the total of phenotypic variation. So additive genotypic variation is a subset of total genotypic variation. But here's the key point. Heritability is a number which tells you how much phenotypic variation is attributable to genotypic variation. Okay, so let's move on. So why are these terms important, and why do we care? Why am I mentioning them and taking the time to devote a third of the lecture to heritability? Well, broad sense heritability is very important in studies in a medical context where you want to understand how much of your propensity to a given disease is due to genetic factors. And that can help biomedical scientists and physicians understand to what extent they'll be able to predict your phenotype from your genotype. Narrow sense heritability is primarily of interest in the context of breeding. So for instance, in agriculture, when you breed animals, farm animals like cows and pigs, and you want a certain type of phenotype, like more meat or more egg production or more milk production, heritability is a very useful measure to help guide your breeding program. Same is true with plants. So that's why we care. Now, we're not going to devote a lot of time to this, and it's not crucial you understand these formula in great detail. But essentially what this says is the variance due to genes can be divided up into three categories. Additive genetic variance, which is the basis for narrow sense heritability. Dominance variation, which is like sickle cell, where you have a recessive gene, and of course that means that the wild type allele, the A as opposed to the S, is dominant. Okay, because you need 2 copies of the S in order to get the phenotype associated with S. And the I refers to epistatic interactions between genes. So you can get variation in genes due to those 3 types of genetic variation. Now, the phenotypic variation overall includes those 3 sources of genetic variation, and then it includes environmental variation, situations where the effect of the environment depends on the genes you have. So for instance, very educated parents are more likely to read to their children. So the environment of children in an educated household may be different than the environment in a non-educated household. So that's an interaction between genes and environment. And then there are sometimes correlations between genes and environment, purely statistical, where there's no necessarily causal arrow in either direction. So now let's talk about heritability and twins. I have to say, these are among the cutest twins I've seen. So Heritability can be estimated in humans by comparing resemblance in the phenotypic traits of twins. And you've all heard of twin studies probably. And they make the following assumptions: that monozygotic or identical twins share all of their genes and their environment, which may or may not always be true. And it certainly isn't necessarily true even prenatally in the uterus. But dizygotic, or fraternal twins share only half their genes and their environment, meaning all their environment, although again, that may not be totally true. But as you know, many times when you hear in the public, in the general press, when you hear reports about how heritable a disease is, it's often in the context of a twin study comparing identical twins to fraternal twins. So here are a couple of quotes. I just want to give you a sense of the kind of ridiculous statements that sometimes get made by people who should know better. So this is from a book by someone who's actually a biomedical investigator and should have known better. So this says, scientists at the Institute for Behavior Genetics in Boulder, Colorado found that 50 to 60% of shyness was inherited in twin babies. Now, that's a meaningless statement. 50% of your shyness, or 60%, whatever the number, cannot be attributed to genes or environment. It's a subtle point, but you can say that 50% of the variation in shyness can be attributed to variation in genotypes. But you can't say that 50% of your shyness is due to genes. Similarly, from this book by Matt Ridley, who's a very well-known science writer, and often thought of as reliable. The conclusion that all these studies converge upon is that about half your IQ was inherited, and less than a fifth was due to the environment you shared with your siblings and the family. Well, again, it isn't that half your IQ is due to genes. It's half of the variation in IQ in a population is attributable to genetic differences among the members of that population. Now, here's a particularly interesting case, which I think will make concrete, will crystallize for you the subtlety of this concept and why the fact that there is an association between a particular gene and a particular phenotype may mislead you about its causal role. So it turns out this is an African who is albino. He has very little melanin in his skin, and it turns out in Africa, particularly in Tanzania and some of the other countries near Tanzania, like Uganda, albinos are terribly discriminated against and persecuted. And in fact, because of various cultural beliefs, they are often physically attacked and literally hacked to death or hacked apart because there are certain assumptions among some members of these societies that body parts from albinos will confer certain magical powers. So their lives and their fitness is much reduced, and it's highly heritable, I presume. I don't know of any study, but this is sort of that's why I labeled it a thought experiment. But the point is, having the homozygous state, which makes you low in melanin, has these incredible consequences. But if you take these people out of that society, out of that cultural context, this gene would have almost none of these implications. So the fact that it's highly heritable does not mean that it's not changeable. So here are some important properties of heritability. It's a property of a population, and it's specific to the environment of that population at that time. It's not a property of a gene, a trait, or an individual. It does not indicate the extent to which genes are involved in creating the trait. It's only about how much phenotypic variation is due to genotypic variation. And this is crucial for understanding the genetic implications the implications of genetic findings in a medical context. The value for the heritability of a trait, like a disease, carries no implications regarding the ability to modify the trait, for example, through environmental alteration, such as medical treatment. So summarizing, genes, most diseases are influenced by both genetic factors and environmental factors. If a disease has a low heritability, one cannot conclude that genes play no role. It depends on what genetic variation is available in that population and what environment they're in. If a disease has a high heritability, one cannot conclude that manipulating the environment will have no beneficial effect in ameliorating disease. Even if my myopia was completely heritable, I can fix it by putting on my glasses. So heritability in one environment doesn't tell you much about heritability in another environment. And the same disease-associated gene can manifest itself quite differently in different individuals. So in conclusion then, diverse evolutionary reasons for there are diverse evolutionary reasons for disease predisposition. Diseases result from genetic and non-genetic factors. The effects of genes on health depend on both genetic and environmental, which is a slightly different point. So diseases clearly result from both kinds of factors, but even the genes themselves, the effects the genes have depend on both other genes and environmental factors. It's not a straight shot from genotype to phenotype. Something's missing there. Heritability is a property of a population, not an individual. And the extent of heritability does not limit the possibility of effective therapy for a disease. So you may be interested to know that in a recent study just published in the past month by a famous researcher from Johns Hopkins, they sequenced many, many genomes of many, many people. And their conclusion was that in many cases, knowing the genotype is not enormously helpful in predicting your disease risk. There are cases where it is predictive, but it isn't always. Anyway, I'm going to stop there, and we can take more questions after we eat dinner.
Yeah.